Hi everyone, welcome to the lecture PowerPoint on muscles and muscle tissue. We will be covering three different types of muscle between AMP1 and even extending into AMP2. However, for this particular lecture on just overall muscle tissue and muscle physiology, skeletal muscle, as noted in the bottom bullet point, will be the primary topic. Skeletal muscle tissue is attached to bones and to skin in certain places. It is what we know as striated. And when you hear the term striated, you want to think striped, right? You can see all these really tiny striations or stripes, a banding pattern, when you look at a micrograph of skeletal muscle tissue. Those striations are actually only seen when you apply a stain, right? When you put some dye on a thin section of tissue. What that is it's a heavy accumulation of dye where the myosin and the actin, the contractile fibers of the muscle, overlap. So you see a dark and then a light. Skeletal muscle is under voluntary control, meaning it's under conscious control. We choose when we want it to contract, and it is extremely powerful. Cardiac muscle tissue is also striated, meaning that you can see visible stripes or striations after staining. It also has a feature where you see these lines in between adjacent cardiac muscle cells, and you'll learn more about those in AMP2. Those are called intercalated discs. And just like you see the striations, which here are really tiny, these are those little teeny stripes here that you see in all the cells. Those intercalated discs, the reason you see those so darkly staining is it's an accumulation of dye where proteins overlap. But cardiac muscle tissue is found only in the heart, and unlike skeletal muscle, it's involuntary, right? We don't choose to have our heart beat. It does that. And then the third type is smooth muscle tissue. And this is found in the walls of hollow organs such as the stomach, the bladder, and your airways, like your trachea, for example. Unlike cardiac and skeletal, smooth muscle is not striated. That is not to say it doesn't also contain contractile proteins, but the manner in which they're arranged does not give those stripes. And like cardiac muscle, smooth muscle is also involuntary. We do not consciously control its contraction. There are certain key characteristics of muscle tissue. So some of the key terms and characteristics you want to be familiar with are: 1, excitability. In regards to muscle tissue, this means the ability to receive and to respond to stimuli. So you can also think of it as responsiveness or irritability. Contractility is the ability to shorten when stimulated, right? So contractility, we're only talking about contracting and shortening. And in opposition to that, we have extensibility, the ability to be stretched. Elasticity is the ability to recoil or recover to resting length after having been stretched. As far as the functions of muscles go, we know that it functions in the movement of bones, right? It acts as levers between bones, or in the case of smooth muscle or cardiac muscle, it's pushing through fluids like blood. 2, maintaining posture and body position. A lot of those paraspinal muscles that we don't really give much thought to, but they're holding us upright. Even if we're sitting in a slouched position, the fact that you're not doubled over onto yourself is due to those muscles always being somewhat contracted. Muscle also functions in stabilizing joints and having us be appropriately positioned in 3D space, and heat generation, especially skeletal muscle. So in a skeletal muscle, each muscle is served by at least one artery, one nerve, and one or more veins. The connective tissue sheaths of skeletal muscle are very strategically arranged, if you will, right? You look at this thing in front of you, and you may have actually seen, like if you've ever sliced flank steak, something that's a kind of a chewy steak, you may actually see these connective tissue separations. So just to put it into reference, right, this is the humerus that you're looking at, the upper arm bone. Here's the tendon, and here's one of your bicep muscles, for example. The epimysium, okay, this layer right here is that thin layer on the outside. It's dense, regular connective tissue, and it surrounds the entire muscle. If you've purchased chicken breast, for example, and had to pick off like that thin film over it, that's the epimysium. The perimysium is the fibrous connective tissue that surrounds fascicles. Okay, so what is encircled here, this whole thing that's being pulled out, is a fascicle. A fascicle is a group of muscle fibers. So the muscle fiber is the thinnest, well, no, it's actually not the last thing, right? This thing right here, this guy is a muscle fiber, and inside are the myofibrils, the bundles of contractile proteins. Okay, so a muscle fiber is one single cell. This thing, let me clear the animations here if I can. Actually, I can't right now. But this whole thing that I'm circling again in blue is a single cell. It has multiple nuclei because it has tons of contractile proteins. It's huge. It's really long too, but that's one cell. Okay, so perimysium are the fibers, are the bundles of connective tissue that surround fascicles. Fascicles being bundles of these muscle fibers. And then endomysium, remember endo is within, is the very fine areolar connective tissue that surrounds every single individual muscle fiber. Okay, so perimysium is going to be this right here, surrounding an entire bundle of muscle fibers, fascicles, where every little pink dot is a muscle fiber, right? So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, about 15 muscle fibers in this one fascicle. And then you have one individual muscle fiber, what's circled here in blue, okay? And that is surrounded by a very, very fine layer of connective tissue called the endomysium. Why does it have to be so fine? Because you need diffusion of things across that muscle cell membrane. Okay, so here's an older figure, but a really good one from Pearson. And it shows the structure and organizational levels of skeletal muscle. So again, it talks about the epimysium being around the entire muscle itself. Okay, and it says right here, a muscle consists of hundreds to thousands of muscle cells. A fascicle is a discrete bundle of muscle cells or muscle fibers. The term muscle cell and muscle fiber can be used interchangeably. And that's surrounded by perimysium. And then each individual muscle cell or muscle fiber has that endomysium around it. So muscles can be either directly attached or indirectly attached. When they're directly attached, that means that the epimysium of the muscle, that outer layer that surrounds the entire muscle, is literally fused to the periosteum, the outer connective layer of bone, or to the perichondrium, in certain cases, of cartilage. So for example, your ribs have intercostal muscles. If you've ever eaten ribs, not to be gross, and you have to like gnaw the muscle off the bone, that's a direct attachment. Indirect attachment are when the connective tissue wrappings extend beyond the muscle as a rope-like tendon, or in cases like between like the radius and the ulna or on the back of the femur, a sheet like what's called an aponeurosis, this big sheet of tendon. Think about the indirect, like if you wrapped a hard candy and then the wrapper, you twirl it on either side. That's what we mean by the indirect. That would be the connective tissue then forming the attachment. So if we look at the microscopic anatomy of a single muscle cell or muscle fiber, it's cylindrical in shape, like a straw, and it can be anywhere from 10 to 100 microns in diameter, and believe it or not, up to 30 centimeters long. In a previous research position that I was in, one of the researchers was actually teasing out skeletal muscle cells from mice and putting them on a force transducer. You couldn't even see it because the dimensions are so thin, but it's 30 centimeters long. That's big. Long. So what are the key characteristics of a single muscle cell or fiber? Having multiple peripheral nuclei. So why? One, it's a huge cell, right? So think about you need multiple command stations, if you will. Why are they pushed off to the side? You can see them here. The nuclei are sort of flattened and pushed off to the side, this whole thing being a single muscle cell or fiber. The nuclei are pushed off to the side because the cytoplasm of the cells is so chock full of the contractile proteins, actin and myosin, that there's really no room for anything else. Many mitochondria. Why? Because muscle, especially skeletal muscle, is such a metabolically active tissue. It's also the reason why we need lots of glycosomes, glycogen storage sacs, right? Because we need to break down glycogen into the usable glucose monomers to burn it for power in the muscle, to burn it for energy, and myoglobin for oxygen storage, because we can't have any true production of lots of ATP, right, without oxygen. And these cells also contain myofibrils, right? Do not confuse fibril, which are intracellular bundles of actin and myosin protein, with myofibers being the entire cell. Sarcoplasmic reticulum, which is modified endoplasmic reticulum specifically for muscle, it's where we store the calcium that's key in muscle contraction, and T-tubules, which are like little manholes that are invaginations or dippings in of the cell membrane, so it carries a electrical conduction into the cell. So what we're seeing here in this slide, the entire thing is a single muscle cell or fiber. That's why it says this is one cell. And this is the endomysium, that very fine, loose areolar connective tissue layer over it. These things, one of which you see pulled out, is a subcellular or intracellular structure known as a myofibril, right? These are the densely packed rod-like elements made up of actin and myosin, the contractile proteins of the skeletal muscle cell. And as we've said in previous slides, they make up about 80% of the volume of the cell, which is why the nuclei, the multiple nuclei that you see, are pushed off to the side, to the periphery, because there's no room for them. The way in which the actin and the myosin long cylindrical proteins are laid over one another is what gives us those striations. So these striations are perfectly aligned, repeating patterns of what we know as dark A bands, and we'll go through what each of these mean, and the lighter I bands. The sarcomere is the smallest contractile unit, so it's the smallest functional unit of a muscle fiber or muscle cell. It is the region, it's defined as the region of a myofibril, right? So the region of one of these long stretches of actin and myosin between two successive Z discs, right? And what these Z discs really are just accumulations of proteins to anchor the thick myosin filament that you happen to see in green, right, in this figure, and then the thinner actin filaments seen in pink. So you'll see the colors change depending on what diagram I've included. Don't pay attention to the colors, pay attention to the band names and where there's overlap, and thick and thin. So thick is myosin, thin is actin. Where we see from one, keep in mind, a sarcomere is from one Z disc to the next. So in this figure, you see, how many sarcomeres do you see, right? 1, 2, 3 sarcomeres. Within a single sarcomere, we have an A band, which is defined as the length of the thick myosin filament, and then the thin filaments make up the I band, right? Now, you can see this thin in this diagram, it's red. That thin actin filament actually spans 2 adjacent sarcomeres. So within a single sarcomere, if we're looking at just this one here on the right-hand side, then the A band, right, is from one end of the thick filament to the next. And within there, you see some overlap with the thin actin filament. Okay, and then you have stretches where it's just the thin actin, that's known as the I band. Okay, so we have that thin filament, and we're going to go over these bands over and over again. The A band is where there is an overlap of thick and thin, except for this area in the middle known as the H zone. That's where there's just the thick myosin. So the A band is the stacked set of thick or myosin filaments along with the portions of the thin or actin filaments that overlap with them. So the two ends of the thick filaments within a stack define one given A band. The I band, as I've noted previously, consists of the remaining portion of the thin actin filaments that do not overlap with the A band, with the myosin. And as I've noted, one thin actin filament, here it's shown in blue, spans more than one sarcomere. This diagram isn't the greatest to show that because they're on different levels, but this is one continuous actin filament. The Z disc is a dense vertical line visible after staining, right, in the middle of each I band. So again, from one Z disc to the next defines one sarcomere, one functional unit of muscle. The H zone is the slightly lighter middle region of that A myosin band, where the thick and thin do not overlap, where it's just the thick. And then the M line is a line of protein known as myomesin, which is where the M comes from, that holds adjacent thick filaments together. It's like little snaps to make sure that the A bands all stay directly stacked on top, right? These are like little snap proteins in the middle. And again, this is just a nice sort of summary figure. We start at the top with a single muscle fiber, a single muscle cell. What's pulled out here is one myofibril, right? One organized group of actin and myosin. And then we can see the various bands. So here we see 3 sarcomeres, right? The one in the middle, you can see each of the Z discs. And then on the bottom, you see the arrangement of those actin and myosin filaments. The thick filament is composed of the protein myosin. Myosin has 2 tails that are interwoven, kind of like twirling around 2 strands of pearls. And these are made of heavy polypeptide chains, 2 heavy protein chains. Each of these chains, here's one right here, right, has a head, this globular head, which contains 2 smaller light polypeptide chains that act as something called cross bridges during muscular contraction. On the myosin heads are binding sites for actin of the thin filaments, binding sites for ATP, cellular energy, as well as ATPase enzymes, which will hydrolyze ATP to fuel the muscular contraction, something called the power stroke. The thin filament, or actin, is, again, kind of like a twisted strand of pearls, with the filamentous strand being referred to as F-actin, and each individual little pearl or globular molecule as a G, or globular actin subunit. Each of those individual globular subunits bears active sites for the myosin head to attach during contraction. Now, we don't want muscle to be able to contract just any time, right? That would lead to potentially us going into tetanus. So we need things to block the actin sites where myosin wants to chomp on when contraction is not supposed to happen. And that's the job of the regulatory proteins troponin and tropomyosin. Think of each of those little individual globular subunits of actin, right? You can see those little dots on them. Those are the binding sites for myosin, for the heads of myosin. So when we do not want muscle to be contracted, this string-like molecule, okay, known as tropomyosin, blocks all of the binding sites of actin. It's like putting a little rope through those little mouths. And then this triple globular molecule called troponin, basically it's like if I put like a tablecloth over a table on top of answer keys for an exam, okay? That prevents you from seeing the answer keys, but you could easily just pull that thing right off the table. So instead, I sit on the tablecloth to make sure that you don't lift it up. Troponin holds the tropomyosin in place. When calcium is released, and we'll learn about that shortly, during muscular contraction, when calcium comes out of the sarcoplasmic reticulum, it binds to troponin, this particular part right here, troponin C of that triple molecule, and basically pulls the troponin and the tropomyosin along with it away from those actin binding sites, and then myosin can latch on. So in the static PowerPoint, I want you to watch these two videos on the cross-bridge cycle, but we'll go through it sequentially from step 1 through step 4. So here in step 1, okay, we see the myosin cross-bridge attaches to the actin myofilament. So preceding this was release of calcium and the rolling off of troponin and tropomyosin. Okay, 2, and this is key, remember the myosin head also has an enzyme site for ATPase enzymes. So the working stroke or the power stroke happens because the myosin head pivots and bends after the ATP is hydrolyzed, because now it's energized, sliding the filament one more notch towards that middle M line, okay, towards the middle of the sarcomere. Then the only thing that's going to cause the myosin head to release and recock so it can do another power stroke is to bind another molecule of ATP. As new ATP attaches to the myosin head, the cross bridge detaches. Okay, and then as ATP is split again, we cock and load that myosin head and another power stroke happens. The sarcoplasmic reticulum, as I mentioned previously, is a modified network of smooth endoplasmic reticulum surrounding each myofibril, okay, each of those bundles of actin and myosin inside of a single cell. So here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9 myofibrils inside of one muscle fiber or muscle cell. And you'll see that all this little kind of teal colored meshwork is sarcoplasmic reticulum enveloping each individual myofibril, and then it has these thickened sacs called the terminal cisternae, and they form perpendicular cross channels. The major function of the sarcoplasmic reticulum is to function in the regulation of intracellular calcium release during muscle contraction. The T-tubules, shown here in pink, right, the sarcoplasmic reticulum, those terminal cisternae, all the sarcoplasmic reticulum in this picture is shown in yellow, okay, with the mesh-like thinner tubules in the middle and then the swellings over here. And then in between each segment of sarcoplasmic reticulum is a T-tubule, which is a dipping in, an invagination of the cell's membrane, known as the sarcolemma, so that electrical signals can penetrate deep into the cell's interior. Remember that muscle fibers, muscle cells, are very, very dense. They don't have a lot of liquid in their cytoplasm. It's 80% the myosin and actin. So to get conduction of a signal into there, it needs to go through this extension of the plasma membrane. Here in this photo, please don't pay attention to the colors necessarily, because those are going to change depending on the diagram. But in the darker blue, you see the sarcoplasmic reticulum, and in the whitish blue, you see the T-tubules. So because it's always one segment of sarcoplasmic reticulum and then a T-tubule sandwiched in between, and then the other terminal cisternae of the adjacent sarcoplasmic reticulum, this little pairing where you have a terminal cisternae, right, the swelling of one sarcoplasmic reticulum sandwiched with a T-tubule and then another sarcoplasmic reticulum, terminal cisternae, this thing right here, it's called a triad. And that's important, right? Because I always think of the T-tubule like a hallway, say like in a kennel, and you have the rows of cages on either side. When calcium is released from those lateral sacs of the terminal cisternae here and here, that's what's going to open voltage-gated channels in the muscle fiber, causing excitation. So it's going to spread down this T-tubule. Okay, so again, the triad is the T-tubule sandwiched between the two terminal cisternae of adjacent segments of sarcoplasmic reticulum. The T-tubules have the job of conducting nervous, aka electrical impulses, deep into the muscle fiber, and the sarcoplasmic reticulum functions in calcium storage and release. So as an action potential, and we have not gotten to this yet, we will, right? But an electrical impulse caused by opening of voltage-gated channels is propagated along the sarcolemma, and then it dips down into the T-tubules. Calcium ions are subsequently opened as that voltage change opens voltage-gated calcium channels in the sarcoplasmic reticulum. Now, how does this translate into contraction? Contraction, by definition, is the generation of force. It does not necessarily cause shortening of a muscle fiber. Shortening occurs when the tension generated by the cross bridge between myosin and actin on the thin filaments exceeds the forces opposing shortening. So in the relaxed state, thin and thick filaments overlap only slightly. During contraction, the myosin heads bind to actin, then detach and bind again, propelling the thin actin filaments over the myosin filaments closer to the M line, the middle of each sarcomere. So what happens on a sarcomere level during contraction? So let's walk through this. 1, the H zone disappears. So what is the H zone? Remember, in relaxed muscle, the H zone is the tiny portion in the middle of the A band where there is only the thick myosin filament, no overlap with the thin actin. This disappears because during contraction we get maximal overlap of actin and myosin. The I band also gets smaller. Remember, the I band is the portion of the thin actin filament that doesn't overlap with myosin. So again, when contraction is occurring, we're increasing overlap of actin and myosin, so the I band also gets smaller. The Z discs, the ends of the sarcomeres, get closer together because that's what we want to do. And the A band is the only thing that does not change in length. Neither actin or myosin filaments shorten, but it's the degree of overlap. But because the A band is the center, the myosin filament does not do any moving, okay? So the A band doesn't shorten. The actin filament moves relative to the myosin. So how does this translate into the banding patterns that you see, the striations? So the darkest part right here is the A band, right? And you see it's darker here and here where there's overlap of actin and myosin. In the middle, where it's just the myosin, it's a little lighter. It is not quite as light as the I band, right, between each sarcomere that's just the thin actin, but you'll see where it's just the thin actin, it's the lightest. Where it's just the myosin, it's a little bit lighter, right, but the myosin is a thicker filament, so it lets less light through. And then where there's overlap of actin and myosin, that's the darkest. I like to give you all views multiple times in different ways. So again, walking through now what's happening at the level of the actin and myosin fibers, translating into what you see under the microscope, right? When it's relaxed, you clearly see the H zone, okay? You see the whole A band and you see the I band, and then as it contracts, the H zone goes away and the I bands get much, much smaller, all because the degree of overlap between actin and myosin increases. What are the requirements for skeletal muscle contraction? 1 is activation. We need neural stimulation at something called the neuromuscular junction, the junction between the nerve ending and the muscle cell. 2nd is excitation-contraction coupling. This involves the generation and propagation or spreading of an action potential, an electrical impulse, along the sarcolemma, aka the muscle cell membrane. The final trigger is a brief rise in intracellular calcium in the muscle fibers. Skeletal muscles are stimulated by somatic motor neurons. Remember that skeletal muscle is under voluntary or conscious control, so it's somatic motor neurons that we can consciously control the firing of. Axons, the transmission parts of these motor neurons, travel from the central nervous system, the brain and spinal cord, via nerves, bundles of axons, to skeletal muscles. Each axon forms several branches at its end as it enters a muscle. Each axon ending forms a neuromuscular junction with a single muscle fiber, a single muscle cell. Now, where the neuron fires onto a muscle cell is usually midway along the length of the muscle fiber, because if you think about a muscle fiber being this long cylindrical multinucleated cell, right, we want to fire onto it, so to speak, right in the middle so that the impulse can travel in both directions simultaneously, speeding it up. The axon terminal and muscle fiber are separated physically by a small fluid-filled space called the synaptic cleft. Synaptic vesicles are released from the axon terminal and they contain the neurotransmitter acetylcholine. The sarcolemma, the muscle cell membrane, happens to contain acetylcholine receptors. When a nerve impulse arrives at the axon terminal, acetylcholine is released from those vesicles and binds with receptors on the muscle cell membrane, the sarcolemma. When these receptors open, they also are ion channels. So it lets in this case, more positively charged sodium into the muscle cell, changing the voltage or changing the electricity of the muscle cell. So here you can see an action potential, right? A change in voltage, meaning a change in ion concentration across the membrane, arrives at the axon terminal. This stimulates calcium to move into that axon terminal, and the entrance of calcium stimulates the release of acetylcholine from the axon of the neuron. This acetylcholine goes across the synaptic cleft and binds to these receptors, which again happen to be ion channels on the muscle cell membrane, letting more positive charge into the muscle cell and causing its excitation. At low intracellular calcium in muscle cells, again, like we mentioned before, tropomyosin and troponin is blocking the binding sites on actin that myosin wants to grab onto. When a muscle cell is excited because ions are entering, the intracellular calcium levels will rise. Calcium will be released from the sarcoplasmic reticulum, and this will cause troponin and tropomyosin to roll off those actin binding sites, allowing myosin to bind. When nervous stimulation is done, the calcium is pumped back into the sarcoplasmic reticulum, ending contraction. Now, the cross-bridge cycle, the binding of the myosin head to the binding sites on actin, will continue as long as calcium is free and in the muscle fiber cytoplasm, and adequate ATP is present. So cross-bridge formation is when that high-energy myosin head attaches to the thin filament, it splits ATP, and then pulls the actin one notch, one stroke, closer to the middle of the sarcomere. ATP, a new molecule of ATP, has to attach to the myosin head in order for it to let go of the actin and then recock and position again. Okay, so here we have cross-bridge formation. The energy for that power stroke is derived from the hydrolysis, the splitting of ATP. Okay, power stroke happens. New molecule of ATP has to come in and cause the release of the myosin head. It recocks, splits it again, another power stroke. The same principles apply to the contraction of a single muscle fiber as it does to a whole muscle. Contraction produces tension. Tension is the force exerted on the load or object to be moved. Contraction does not always shorten a muscle. There are isometric contractions and isotonic. Iso means the same or equal. Metric refers to the length, and tonic refers to the level of tension. So isometric would be the same length, meaning the muscle doesn't change in length. Isotonic means that the level of tension is the same throughout. Isotonic contractions, the tension generated by the muscle is greater than the force exerted on the load, by the load on the muscle. So the result is that the muscle changes in length and moves the load. Notice that it's the tension, right? The tonic means tension in this case that stays the same, not the muscle length. In this case, the muscle length changes and shortens and moves the load. In an isometric contraction, the load exerted on the muscle is greater than the tension that the muscle is able to develop. Therefore, the tension increases to the muscle's capacity, but the muscle neither shortens nor lengthens. And the force and duration of contraction vary in response to stimuli of different frequencies and different intensities. A motor unit is a single motor neuron, and all could be anywhere from 4 to several hundred, all of the muscle fibers that it supplies. So here we have motor unit 1 supplying 2 muscle fibers and motor unit 2 supplying 3. So in this case, motor unit 3 is larger than motor unit 1.